Good morning, everyone. It is my honor to introduce Dr. Sally Radovick. Dr. Radovick is the Henry Rutgers Term Chair and Professor of Pediatrics and Senior Associate Dean for Clinical and Translational Research at Rutgers Robert Wood Johnson Medical School. Prior to this position, she was the Division Director for Pediatric Endocrinology and the Vice Chair for Research in the Department of Pediatrics at Johns Hopkins University. She received her medical degree from Northeastern Ohio University's College of Medicine and completed her residency in pediatrics at Case Western Reserve. She completed her fellowship in pediatric endocrinology at the NIH. Dr. Radovick is a specialist in growth and development and the pubertal disorders in children. Her research is focused on determining the regulation of the gonadotropin-releasing hormone gene, which is central in the role of controlling the onset of puberty. She's authored or co-authored more than 100 peer-reviewed articles and has been invited to write a dozen book chapters in her field. She serves as the Associate Editor for Pediatric Endocrinology, the Editor of Pituitary, and the Editor in the Current Opinion of Pediatric Endocrinology. Dr. Radovick serves as the President of the Pediatric Endocrine Society and the Chair of the Research Council and a Council member and the Chair of the Drug and Therapeutics Committee. She's a member of the Guidelines Committee and the Finance Committee for the Endocrine Society. She's also a co-author of the AACE Growth Hormone Guidelines. Dr. Radovick has trained over 75 postdoctoral students, postdoctoral fellows, and junior faculty in scientific research, and is the PI of the Rutgers CTSI KL2 program. She has remained clinically active during her career and focuses on children with disorders of growth and puberty and precepting teaching clinics in pediatric endocrinology. Please join me in welcoming Dr. Sally Radovick.
Good morning, and thank you for that very kind introduction. I'm very excited to be here because I've waited a year for this, more than a year actually for this. And unfortunately, it still has not come to pass that I could be there in person. So thank you for bearing with this and inviting me back. So let me start by sharing my screen. All right, how's that? Will someone, is it good? All right, let's go. All right, today my discussion will focus on Kispeptin, and I will try to convince you that Kispeptin is the missing link between puberty, reproduction, and metabolism. I have no conflicts of interest. In order to accomplish our objectives, we will look at Kispeptin in 5 different ways in its multiple physiologic functions. So its function in reproduction. I will show you that Kispeptin is a recently identified neural protein that provides the major signal for pubertal onset and reproductive development. That Kispeptin is secreted from the arcuate nucleus and controls the estrous cycle. That and I will discuss the role of Kispeptin in puberty and implicate the estrogen receptor alpha in the control of pubertal onset. And lastly, show some interesting data about Kispeptin, which is found in the liver and acts as a metabolic factor. All right, so we'll start with the reproductive axis, which is familiar to many of you, but let's just do a quick review of the function of the reproductive axis. So reproduction is controlled centrally in the hypothalamus by Kispeptin, which is a newly found protein that is secreted in 2 nuclei in the hypothalamus. And there are Kispeptin receptors on GnRH neurons that respond to bind Kispeptin, respond to Kispeptin, and result in the synthesis and secretion of pulsatile GnRH. The pulsatility of GnRH is absolutely critical for normal reproduction. Pulsatile GnRH then is released into the portal system between the to the pituitary gland and results in the synthesis and secretion of luteinizing hormone, LH, and follicle stimulating hormone. For every pulse of GnRH, there is an equivalent pulse of LH, and that is responsible for sex steroid synthesis by the gonad. FSH is also controlled by GnRH, but is also responsible to other, especially ovarian and testicular steroids. There is a very complex feedback mechanism that is tightly controlled in the axis, such that sex steroids feedback at both the level of the pituitary at the gonadotrope, as well as at the level of the brain to control synthesis and secretion of this of the proteins in this axis. In most of the time, in both males and females, control is negative, so that sex steroids feedback to decrease the synthesis and secretion of Kispeptin, GnRH, and gonadotropins. However, positive feedback is only seen in females and is critical for the pre-ovulatory surge that controls ovulation. Okay. A very seminal contribution to the field was made by two laboratories that showed that patients who had mutations in the Kispeptin receptor had hypogonadism. This was the beginning of the investigation of Kispeptin having a role in neuroendocrine control of puberty and reproduction. However, interestingly, Kispeptin was actually first discovered as metastatin inhibitory factor, MIF. It was found by cancer biologists actually in Hershey, Pennsylvania. And they showed that Kispeptin, or what they called metastatin, was an inhibitory factor for metastases, but it wasn't very good at that. And so they more or less abandoned the study of metastatin. And not until this protein was found to have a role in reproduction did it again begin to be studied. And at that time, it was called Kispeptin because it was discovered in Hershey, the Hershey Kiss, Pennsylvania, hence the name. Following that, there were patients who had mutations in the ligand Kispeptin who were also found to be hypogonadal. So Kispeptin started to take on even more of an important role. As you can imagine, if there's a push, there's a pull, and not only were there mutations found in the system that resulted in hypogonadism, but a mutation in the receptor was found in a patient that had central precocious puberty. All right, this is taking coals to Newcastle. This is from Tony Plant's lab, but I think it's an excellent way to introduce Kispeptin. Kispeptin is shown here using immunostaining for in the arcuate nucleus, showing in green Kispeptin and fibers of GnRH in the median eminence. You could see they're very intimate connections, I just think this is a lovely diagram of Kispeptin and exemplifies where Kispeptin is, at least one of the areas that Kispeptin is, and how intimately related it is to GnRH. So Kispeptin is encoded by the Kispeptin gene. It has 4 exons, exon 3, exon 4, encode the messenger RNA. There are multiple splice sites in Kispeptin, and the protein is spliced so that Kispeptin 10 is an active protein, but the active protein is actually can be found down to a decapeptide, a 10 amino acid protein referred to as Kispeptin 10. When I'm discussing Kispeptin today, I will be mostly referring to Kispeptin 54, unless I will discuss specifically Kispeptin 10. The Kispeptin receptor has been cloned. It is a 7 transmembrane spanning receptor. It has a extracellular amino terminus and intracellular carboxy terminus. It is was originally referred to as GPR54 and as an orphan receptor, and then it was identified to bind Kispeptin. It's a member of the galanin family of receptors. It signals through the G sub Q pathway. It increases intracellular calcium levels and calcium-dependent signaling. It activates protein kinase C and the MAP kinase pathway. Kispeptin is expressed in a variety of tissues in addition to the brain. It has been found in liver, adipocytes, the spleen, the testes, the placenta, and the ovary. So you can just imagine that it may actually have a role beyond the neuroendocrine system, and what those roles may be in these multiple tissues where it's been found not only in the RNA level, but at the protein level, for many tissues remains unknown. We will spend some time today discussing its role in the liver. The receptor, the Kispeptin receptor, is also found in some of these tissues, and how the relationship of Kispeptin to its receptor isn't clear. So in humans, Kispeptin in blood can be measured in plasma. There is an immunoassay now for Kispeptin, and it's been shown that it varies with the human menstrual cycle, so that a high level of Kispeptin is seen prior to ovulation, very similar to the peak in luteinizing hormone, which signals ovulation, and that it may have a relationship to both estradiol and progesterone secretion, because it is high when estradiol is low, and then has a secondary peak when progesterone levels are elevated. So despite the fact that there are known mutations there was an excitement about the fact that there are translational effects of Kispeptin, there's still an unclear knowledge about how hypogonadism results from mutations in either Kispeptin or the Kispeptin receptor. And so I want to begin my talk by sharing with you some of the data from our laboratory that will give some understanding to the mechanistic effects. So first, just to summarize what was actually many years of research for you, is to think about the neuroendocrine axis in terms of Kispeptin and GnRH, which I showed you very nicely in Tony Plant's diagram, and tell you that when we started our studies, we first looked at the GnRH gene to determine whether there was any direct effect of Kispeptin on GnRH itself. So in both in vitro, which is how we began, as well as later in vivo in mouse models, we showed that the GnRH neuron has Kispeptin receptors that are located on the cell body, as well as on terminals, that binding of Kispeptin to GnRH results in the expression of GnRH. We showed that this is transcriptional, that binding of Kispeptin to its receptor results in the expression of a protein referred to as OTX2, which is a homeodomain protein, which we previously showed was important for both development of the GnRH neuron as well as for activation and expression of GnRH. The OTX2 binding site on the GnRH promoter takes place in an area referred to as the enhancer, and that relates to the mechanism by which OTX2 can increase the expression of GnRH. And also that activation of the Kispeptin receptor on GnRH cells is dose dependent. So although we clearly had some idea about the mechanism that Kispeptin used to activate GnRH in vitro and in some transgenic models, we certainly didn't understand how this may affect reproduction. So we went ahead to try to understand how the Kispeptin receptor on GnRH neurons controlled reproduction. This is a schematic of a knockout mouse model that we made to try to understand the mechanism of the signaling by the Kispeptin receptor on GnRH. So on the left is the diagram of a transgenic mouse. This mouse carries the promoter of GnRH that targets only GnRH neurons. It was fused genetically to Cre recombinase. The second mouse is a mouse that contains the Kispeptin receptor that has had genetically inserted LOX-P sites on either side of exon 3, which is a coding exon for the Kispeptin receptor, so that the mating of these mice results in the following mouse. On all tissues, the mouse expresses the Kispeptin phloxed, which is a normal mouse. It just happens to have the Kispeptin receptor that has LOX-P sites. However, in mice that carry the GnRH Cre, which is only expressed in GnRH neurons, the Kispeptin receptor is deleted in those GnRH neurons where the Cre recombinase recombines the LOX-P sites. Hence, the Kispeptin receptor is deleted from GnRH neurons. This allows us to understand how the Kispeptin receptor functions in not transmitting its signal to GnRH. So this is a result of these studies, and I'll show you these mice. These are females on the left, males on the right. Both of these mouse models are hypogonadal, and I'll show you evidence of that. They have small genitalia. In the female, you can see the fallopian tubes are hypoplastic. The uterus is also small. Shown are the ovaries. On the left is the normal wild-type adult ovary. It contains corpora lutea, which are luteinized follicles that result from ovulation. So this is an excellent way in a mouse model to determine that ovulation has occurred. And in fact, in this model, there's a pre-ovulatory follicle that is on its way to becoming luteinized. On the right is the knockout model that we call GKIRCO, or GnRH Kispeptin receptor knockout mouse. And this is the ovary from that mouse. There are no corpora lutea present, which indicates that there are no ovulatory events. And there are follicles present. However, they are very early follicles, preantral, primary, and antral. And there is lack of progression of a normal follicle development. On the right are males. You can see from the diagram that's dissected testes that the testes are very small. The weights are shown in the graph on the right. We looked at seminiferous tubules and the epididymis, and you can see that in wild type mice, there's sperm present in the seminiferous tubules. However, if you look on the right, there's no evidence either in the seminiferous tubules or the epididymis that there is the presence of sperm. However, GnRH neurons are perfectly normal. This is a view again of the third ventricle, and staining for GnRH. You can see cell bodies of GnRH and their axons. And so we counted GnRH neurons. There was no change in distribution or numbers of GnRH. We looked at puberty in these mice because we wanted to know what the effect of the GnRH receptor, of the Kispeptin receptor was on GnRH neurons in terms of pubertal onset and development. As you can see on the left in females, in the diagram, we looked at vaginal opening. Vaginal opening in mice is the first sign of pubertal onset. In humans, we look at the onset of breast development. In mice, we look at vaginal opening. In females, and you could see from the diagram that in our colony, most mice will have puberty by the time they are, for sure, a month old. Usually in the late 20 days of life, we will see vaginal opening in our colony. You could see that this is significantly delayed or doesn't occur at all in females who have the deleted GnRH, the deleted Kispeptin receptor in GnRH neurons. On the right are males. And again, we look at preputial separation, which is a testosterone effect. In males, in human males, we look at testicular size. In mice, we look at preputial separation and we measure that. And you could see that males also have delayed or lack of pubertal onset. It's very important to look at body weight because there are intimate relationships between body weight and pubertal onset. With this genetic disruption, there was no change or difference in body weight between the knockout mice and the control wild type mice. We looked at estrous cycling. So instead of the menstrual cycle in mice, we have the estrous cycle. We look at vaginal washings from mice, and under the microscope, we look for evidence of estrus, proestrus, or metaestrus, diestrus. And on the what you'll see in the graphs is movement of the epithelium from a cornified epithelium, which signifies the estrous cycle, through the cycles of cell type changes, and which happens normally on the left. Cycles in mice occur, anywhere from 5 to 7 days, so that in a 12-day period, as you can see here, we could usually see about 2 cycles that have happened. And in the knockout mice, we see mostly mice in the diestrus or metaestrus phase. We looked at gonadotropins in these mice, and as you can imagine, both LH and FSH were low. Shown on the left is LH and FSH in females from the wild type and the knockout mice. And similarly on the right is LH and FSH from the wild type and knockout mice. We looked at fertility, and this is just schematically showing fertility in these mice. So our mating scheme is that we take either wild type or knockout females and introduce them to a cage that has an experienced male, or we take similarly males that are either wild type or knockout, and then introduce them to a cage with a female who's been, for wild type, previously pregnant. We could see here that when we mate a wild type mice, they always get pregnant. When we mate the knockout females with wild type mice, we get no pregnancies. Similarly, when we mate the wild type females with the knockout males, we get no pregnancies. So both males and females are infertile. So just to summarize the results of these studies, so Kisspeptin is increased Kisspeptin increases the synthesis and secretion of GnRH. There is a Kisspeptin response element in the enhancer of the GnRH promoter that responds to OTX2. Deletion of the Kispeptin receptor in GnRH neurons in vivo results in perfectly normal GnRH neurons, a delay or lack of puberty, hypogonadotropic hypogonadism with low LH, low FSH, and disrupted estrous cycle, and male and female infertility. So we're going to make this a little more complicated because there are actually two nuclei in the hypothalamus that have Kispeptin neurons, and I've shown them here diagrammatically. Kispeptin cells are located in the arcuate nucleus as well as in the AVPV. This is the anteroventral paraventricular nucleus. It is both of these nuclei have Kispeptin cells. The Kispeptin cells from both of these nuclei signal to GnRH neurons and GnRH responds with synthesis and secretion of GnRH. The difference in these cells, one of the differences in these cells is their responsiveness to estradiol. So arcuate Kispeptin neurons respond with negative regulation to estradiol, while Kispeptin cells in the AVPV respond with increased synthesis and secretion after estradiol stimulus. So the other difference is that arcuate Kispeptin cells secrete other proteins, and they're called candy cells. I know, very cute. But the reason they're called candy cells is that they make, in addition to Kispeptin, neurokinin B and polydynorphin. And this is very important because it helps with understanding the characteristics of this cell type and what it does. And also is going to become a tool for us to study this arcuate Kispeptin neuron further. There have been many studies that have shown that there are different mechanisms by which these cell types work. And only one of those has been to look at gonadectomy and estrogen replacement and feedback from estradiol. However, other studies have begun to characterize the specific roles that these two cell types may have. And there have been some interesting early studies that have used toxin ablation of candy neurons or viral ablation of candy neurons to show that they interfered with feeding behavior, cyclicity. However, these studies were fraught with many problems, including mistargeting or destruction of an entire cell type. So we feel that there's much further to go in trying to understand both of these cell types. And so I'm going to present some of our data that we started to do looking at the candy neuron and its role in reproduction. So this is the title of the next part of the talk. And again, this is the model system that we're going to use to do these studies. And this is the Kiko mouse. The Kiko mouse is a deletion of Kispeptin in just the arcuate nucleus. So we've gone from the GnRH neurons studying the Kispeptin receptor, and now working backwards in the hypothalamus to study the role of Kispeptin, and the ligand and its role in the arcuate nucleus. So again, I've talked about the candy neuron expressing other proteins, and one of those proteins is dynorphin or polydynorphin. Polydynorphin is expressed in virtually all arcuate Kispeptin cells. However, it is not expressed at the protein level in Kispeptin cells anywhere else, which is very important as we try to target just those arcuate cells. So we use that knowledge to develop a mouse model where we could delete Kispeptin only in those polydynorphin candy neurons. So Brad Lowell had previously made the polydynorphin Cree mouse, and we obtained that from him. And we made our own Kispeptin floxed mouse. So again, this is the Kispeptin gene. This is exon 3 of Kispeptin, which is one of the coding exons that was placed between LOXP sites. So again, all of our mice in all of their tissues had the Kispeptin gene that bore LOXP sites. They expressed Kispeptin perfectly normally, except when they were mated with mice that had polydynorphin Cree expressed in, obviously, only in arcuate Kispeptin neurons. So these arcuate Kispeptin neurons, or candy neurons, did not contain Kispeptin. What happens to those mice? All right, so this is, all of this is unpublished data. So I will just show you briefly evidence for the fact that this was knocked out. This is on the left, female arcuate nucleus and male arcuate nucleus. This is immunostaining. In the blue is DAPI that shows you nuclei. In green is KISS1. So the wild type or the top screen, the knockout polydynorphin Cree Kiss flox mice. So what you could see in high power view with the arrows are typical Kispeptin cells. You can see the nucleus with the green Kispeptin. And again, in the male arcuate, you can see similarly, I hope this is coming through on your screens, Kispeptin expression in the male arcuate. The bottom screens, again, at low power, you can see that there is potentially some Kispeptin, but nothing that appears to be in a cell. So there is essentially no Kispeptin expression in the arcuate nucleus. So one of the important things, again, was to measure body weight, because as I said, there is this intimate relationship between body weight and reproduction. And the area around the Kispeptin candy cells has cell types that are responsible probably for appetite, metabolism. However, disruption of just Kispeptin in these neurons had absolutely no effect on body weight. So what happens to puberty? Interestingly, there was absolutely no effect of puberty when we deleted Kispeptin from candy neurons. So on the left, you could see female puberty, pubertal onset, on the right in males. Again, vaginal opening in females and preputial separation in males is used to measure those. between the wild type and their knockout. There was no difference. Again, they all had that. For females, vaginal opening occurred before they were 30 days old, and preputial separation occurred similarly in males. In females, we looked also at first estrus, which is their first cycle. In males, we looked at androgenital distance. Again, absolutely no difference. So the deletion of Kispeptin in the arcuate had absolutely no effect on pubertal onset. The next thing we looked at was Estrous Cyclicity. Again, we did vaginal washings, looked at estrus, proestrus, diestrus, and we've seen cycling. This is 15 days. We get 2.5 cycles in that period of time. In the knockout mice, there may have been some evidence of a cycle every now and then, but essentially they spent most of their time in either diestrus or metastrus, and that's shown in the graph on the right, where there was a decrease of time spent in estrus and proestrus, but a significant increase in time spent in diestrus or metastrus, essentially telling us that the cycle was they weren't cycling, it was disrupted. So I want to talk a little bit about why looking at cycles is important. And again, the University of Pittsburgh and your endocrine group is well known for their studies of a cycle, especially in obese metabolic states. But let me just briefly review that. So the cycle is extremely important. About 25% of all reproductive dysfunction is due to dysfunctions in the cycle. And what you can see is that in high LH pulsatile states, pathophysiology that's associated with those are disorders such as polycystic ovary syndrome, precocious puberty, or even menopause. Low pulsatility states are associated with hypothalamic amenorrhea or obesity and type 2 diabetes. So certainly understanding the cycle, understanding what the molecular proteins are that are important for determining cycle is critical to understanding the physiology of puberty and reproduction. So how do we understand the cycle in mice? So we have to be able to measure the LH pulse. And until only recently, we haven't been able to do that. We can measure basal LH, we can measure stimulated LH, but in mouse models, we were unable to measure the pulse. Fortunately, a very sensitive LH assay has been developed and is being used by a core laboratory, an NICHD core laboratory at the University of Virginia. And so we took advantage of the fact that this was a highly sensitive assay and that it could use very small amounts of blood. About 5 microliters of whole blood could be used in their assay to measure LH. So we are now able to use LH in mouse models to determine the cycle. And so we did this with this mouse model. We took unrestrained, freely moving mice that were acclimated to the handler, who is one of our postdocs, and every 6 minutes for 3 hours, extracted whole blood from the tail vein of the mouse that was nicked, to put that into some saline assay buffer and send it for assay. So let me show you the results of these studies. So I will just first show you a normal cycle in a female mouse on the top. So these are just 2 examples. This is the 3-hour time period that LH is being measured. And what you can see is that there are starred are peaks in the cycle. We measure a peak as an increase of 20% above the baseline, and then a decrease for the next measurement of 20%. And so there are multiple there are multiple tools to use to determine what a pulse is, but we believe that the 20% stringency that we use is very valid. So a typical mouse, female mouse, has 5 peaks in LH during this 3-hour period of time. And you could see that there is a pulse amplitude that's variable, however, ranges anywhere from 0.5 to 0.9 or almost 1. So and then below again is the pulsatile study that was done in the mice that don't have Kispeptin in the arcuate nucleus. You could obviously see that occasionally we do have what's defined as a pulse. However, the mice have very, very low pulse, basal pulse. They have obviously less pulses than the wild type, and their amplitude is diminished. So we could clearly see that there is a role in females for arcuate Kispeptin in mediating pulsatile LH. That's shown in the bar graphs on the right as having an effect on pulsatility and amplitude. These are males. Yes, males pulse too. And shown on the top figures are some examples of wild type mice. And again, starred are the pulses over a 3-hour period in the knockout males. Again, similar to females, they have a pulse every now and then, or what we define as a pulse, but for essentially have very little in the way of pulsatility. Shown on the right is the decrease in frequency, a decrease in amplitude over large numbers there. It wasn't significant, but clearly you could see that there was an effect. So what happens to the gonads with this abnormal pulsatility? So let me show you male and female gonads here shown is the ovary. Again, on the left is corpora lutea from the wild type ovary. On the right, the knockout ovary. Again, a few early follicles in the knockout, but deletion of Kispeptin from the arcuate has resulted in a lack of corpora lutea. It also resulted in, as you can see below, that their ovaries were small. There was a decrease in weight, a decrease in area, as well as total counts for corpora lutea. So in males, again, we saw testicular hypoplasia in the knockouts. See the diagrams on the left. Again, you see sperm follicles. We don't see any sperm in most of the follicles. However, every now and then it does appear that there is some sperm production. The testes are small, they weigh less, they have a lesser area. But there is some evidence of sperm production in some of these males. What happens to we saw LH, what happens to FSH? Again, FSH decreases in the knockout mice, so in both males and females. And we did our fertility study. And let me show you the interesting fertility study. Again, similar to what you saw previously, when you delete Kispeptin from the arcuate, you get infertility in the females. So all of our females who are knockouts are infertile. However, the males have some variability. So the knockout males will occasionally get a female pregnant. There's variability amongst the males. Some are completely infertile, others will perform, at least early on, with some evidence of being able to impregnate a female. So we can summarize by saying candy neurons in the arcuate nucleus are responsible for estrous cycling. For normal release of LH and gametogenesis, but they're not important for the onset of puberty, and they're not important for the control of body weight, and that the effect on reproduction is infertility in females and variable or subfertility in the male mice. All right, so we talked already about Kispeptin and arcuate neurons, and we talked about the role of the estrogen receptor conveying estrogen signals to the arcuate and to the AVP Kispeptin cells. So one of the next tasks we had for ourselves is to determine what that role of estrogen is in regulating Kispeptin. Again, we're going back to this mechanism that we use as our model for study. This time, obviously, different mice. We're going to target Kispeptin cells, obviously, to look at Kispeptin in these two nuclei. And we're going to target the estrogen. We're going to target estrogen feedback. And we're going to do that by deleting the estrogen receptor from those neurons. And again, on the left, we have Kispeptin that is fused to the Cre recombinase. On the right, we have exon 2 of ER alpha that our laboratory has, quote, floxed, and in tissues perfectly normal to have estrogen receptor expressed everywhere except in the Kispeptin neurons where estrogen receptor will be deleted. So what happens? So let me tell you, this is one of those things that happens every now and then. We saw immediately that vaginal opening in this mouse model was early. So as you saw previously in our colony and in these mice models, their controls, puberty or vaginal opening in females occurred about 28 days or so, but for sure by the time they were 30 days old, indeed that's happening. But when we deleted the estrogen receptor from Kispeptin cells, the age of vaginal opening was about half that. Almost all of the mice had vaginal opening by the time they were 15 days old. We looked at LH in those mice. Indeed, by the time they were 15, they had elevated LH levels, and it persisted by the time they were 25 days old, while other mice, obviously at those ages, did not have increase in LH. So what's happening here? And I'd like to spend a couple minutes talking about this slide and the hypothesis that we have. So there's the model for the control of the onset of puberty, posits that Kispeptin has an important role, and it's critical to this neural machinery that's essential for generating not only pulsatile GnRH, which you've seen, but this is important during puberty. So in the left panel during infancy, arcuate GnRH pulse generation is robust. This is, some people refer to this as mini puberty, which really is a misnomer. This is neonatal activation of the reproductive axis. It occurs normally. It's robust. There's intermittent release of Kispeptin, GnRH, LH, and FSH that can actually be measured. And there's a transition then from infancy to the what's called prepubertal or the juvenile stage in the middle panel here, where there is a neurological break shown in red. So this break that is now is holding this arcuate GnRH pulsatile mechanism in check so that There's suppression of Kispeptin, suppression of GnRH, LH, and FSH during this prepubertal juvenile phase. And then puberty becomes triggered when this brake is removed. And GnRH pulsatility begins again, controlled by Kispeptin, we believe. And the entire mystery of what is puberty and what turns it on and off is related to this brake. And we now, from these studies, believe that this brake involves Kispeptin. This brake obviously involves estradiol, and that the ER alpha mediating the estradiol effect in Kispeptin neurons is responsible for this suppression of the pubertal axis in this juvenile or prepubertal break-on phase. And then the release of that suppression by estradiol, which we believe is part of the brake mechanism, results then in pubertal activation. So the last part of this, our discussion will be on metabolism and the role of hepatic Kispeptin. So I'd like to first start by talking about the metabolic system, the peripheral metabolic system, and Kispeptin's role potentially as a liver protein in that, and how our studies got us to focus on this area. So this is just diagrammatically showing you an obese state and the metabolic function during that obese state. So in obesity, there's increase in glucose, but there's also insulin resistance. So the insulin that's secreted in relatively high amounts from the pancreas meets resistance, and that resistance results in the increase in both lack of glucose uptake peripherally, as well as very, very large amounts of glucose that are put out by the liver. Fueling this fire is glucagon, which is also appears to be released when insulin is at high levels, and fuels the fire of gluconeogenesis in the liver. I was working with a colleague who was interested in this interesting glucose, this glucagon mechanism, and why it was released, how it functioned in the liver to turn on gluconeogenesis, and whether there was some signal from this pathway in the liver during this hyper glucose state to the pancreas. So let me show you very briefly. This is another mouse model. It's not mine. This was actually, many people have made modifications on this mouse model. The latest was the Stratakis lab. But what it is a mouse model that tries to mimic that glucagon pathway. So glucagon goes to the liver, increases the cyclic AMP signaling pathway to increase gluconeogenesis. That's well known. So this is a mouse model where this pathway that is used by glucagon is activated, and it's constitutively activated. So, and the way they do this is that with knowledge that it's the PKA signaling pathway. And so this signaling pathway is activated in this mouse model in the liver. So this is a mouse model that has a regulatory subunit of PKA that is between lock sites. And there's a tail vein injection that's done that targets the liver so that this regulatory subunit is released. And what happens when that's released is constitutive activation. And so these mice that have this, that bear this deletion make great amounts of glucose. They are very hyperglycemic. And so my colleague was studying these mice and was looking at proteins that are expressed in these mice. And this is and he did a array analysis, and this is his volcano plot, and he showed me Kisspeptin. So we obviously had known that there was some Kispeptin in the liver, but never had taken it that seriously. So we had the tools to figure this out. We had primers to look at messenger RNA, and we had the Kispeptin antibody to look for protein. So we did that, although didn't ever think that we were going to go down this pathway. And indeed, shown on the right, that indeed these mice have very large amounts of Kispeptin. And these mice also have large amounts of both messenger RNA and protein for Kispeptin. So why is that? Why is Kispeptin high in this hyperglycemic state, activated by the PKA pathway? And in fact, what's it doing in the liver at all? So this is Mehboob Hussain, his laboratory, with Jimmy Song, his postdoc, went ahead and showed that in mouse models of obesity, there was an increase in Kispeptin, similar to what we saw in this knockout mouse model. And in high-fat diet, there was an increase in Kispeptin, messenger RNA, and protein, similar for the leptin receptor knockout mouse. This is the DB mouse. It's just an extremely obese mouse that lacks the leptin receptor. And that there was that this is Kispeptin was a secreted protein from the liver. It was secreted in higher amounts in this high-fat diet mouse, and as well as in this DB mouse. Interesting. Went on in his laboratory to show that in human liver, there was also Kispeptin, so there's human relevance or translation here, and that he could measure Kispeptin, it being high in patients who had type 2 diabetes. So we thought this probably deserved some attention, and we went down that pathway. So I've already shown you the Kispeptin knockout mouse, where we floxed exon 3 of Kispeptin. We used that mouse again, and we did exactly what the lab had done previously. We injected a adenoviral Cree to target the liver, and what you can see is that we deleted Kispeptin from the liver to try to understand what it was doing there. We showed by using messenger RNA and protein that it's decreased, but also we showed that in the plasma, there was a significant decrease in Kispeptin in both males and females, as shown on the right. In fact, about 80% of circulating Kispeptin comes from the liver. So obviously this continued to pique our interest. We looked at a glucose tolerance test in our Kispeptin deletion mice, and they had glucose intolerance. You can see on the left the males, and on the right the glucose tolerance test and the insulin tolerance test. Both males and females had glucose intolerance. They had normal ITTs. They did not have any. These mice, I'll show you later, did not have evidence of insulin resistance. So what's happening with insulin? So when we deleted hepatic Kispeptin, what you can see is that we had an increase in blood glucose, and we had a decrease in blood insulin. So this is referred to as glucose-stimulated insulin secretion. Glucose is injected, insulin is measured at the same time as glucose. So both males and females had decreases in their glucose-stimulated insulin secretion. And we saw an interesting phenomenon that there was a disruption in body weight. So both the males and the females at about 14 weeks of age, began to show a decrease, a significant decrease in body weight. And so we looked at energy, wanted to look at energy expenditure, and we do that using something called the CLAMS, or an indirect calorimetry analysis. And this is our unit on the left. What's important to see here is that there was an increase. This is just male data. Female data is exactly the same. What you can see is that there is an increase in energy expenditure in both the light phase and the dark phase in mice. And this energy expenditure probably explains the decrease in weight that we were seeing. Along with that, there was a decrease in fat mass, as shown in this slide. I'm showing you both males and females. We looked at the decrease in weight in males as well as in females. They had a decrease in fat mass. It didn't affect their lean body mass, again, reflecting their decrease in weight. What about reproduction in these mice? So they reproduce just fine. They have absolutely normal reproductive cycles. So we could say that hepatic Kispeptin is responsible for the majority of circulating insulin. It results in a decrease in insulin secretion, elevations in glucose with glucose intolerance, a change in energy balance with an increase in energy expenditure and a decrease in weight. But hepatic Kispeptin has absolutely no effect on the control of reproduction. All right, so a lot of work went into this from many of my colleagues and my postdocs right now. Baha Likwali, Sarmed Al-Samaria, and Misha Nandakar and Ariel Negwan are working in my lab right now, but many of my colleagues, any of you know John Levine, who is one of the pioneers in Kispeptin research, who I've shared a grant with for almost 20 years now, and much appreciate all the collaboration with him and my funding sources, and of course the Ligand Lab at the University of Virginia. Thank you very much for your attention. I sincerely appreciate being invited, given the opportunity to present this data. And actually, if you think about it, given an additional year to get more data, and the pleasure of being able to at least virtually meet with you. Thank you.
Thank you so much for that fascinating talk. That was great. We've run out a little bit of time, but we're going to have one question, if that's okay. So from one of our adolescent faculty, Dr. Tanya Sayed, said, thank you so much for that very interesting grand rounds. I was wondering about the expression of adipose tissue as it might relate to fertility. I saw the data you showed of no change with body weight, but did that account for adipose tissue?
So in our model of liver deletion of Kispeptin, there was a decrease in adipose tissue. Most of it was subcutaneous tissue. Most of it was subcutaneous adipose tissue. Interestingly, though, we have a colleague who's looking at Kispeptin that's expressed in adipose tissue. And interesting, we just had the last 3 days have been the Kispeptin meetings. Yes, indeed, we do have Kispeptin meetings. There was an abstract that was presented from their laboratory that showed that a deletion of Kispeptin in adipocytes also had a metabolic effect, so that there was some changes in energy expenditure, very little decrease in circulating Kispeptin, at least in a non-perturbed model, and no effect on insulin. So there may be a role for Kispeptin in the liver that's perhaps autocrine.
Great. Well, thank you so much. Thank you everyone for joining us. Please join us later today for our entire day of our Pediatric Discovery Day. We are super excited to have it continuing. And thank you so much. We really appreciate you joining us, Dr. Radovick.
Thank you.